Нет проблем.
Нет проблем.
Нет. Do I sound still like I'm from the Caucasus?
You sound like you're constipated.
Нет проблем. I can imitate a constipated Russian. That's got to be the same.
Actually, opening today's conversation, I combine them, so we're going to pick up from where we stopped yesterday. What's it? Do you have this file on Google? Degradation. So degradation lecture, but it also has copied onto it parts on sorting that we need to discuss and taken from the other presentation. Okay, so good afternoon. Thanks to all of those who came, even though the hour and you're tired and everything. Never mind. So we're going to talk about protein sorting now, and that's going to wrap up our discussion on protein synthesis and traffic. And it's going to be a nice corridor before we get into the discussion on protein degradation. So this was the scheme of our cell, and basically what we're going to be talking now for the next maybe 15 minutes or so is about lysosomal targeting, which takes place in the Golgi, and the decision as to which proteins go to the lysosomes. So we have different kinds of sorting signals which we discussed during class. We talked about the KDL sequence, and we talked about the signal peptide and the concept of having a signal that directs targeting to a particular organelle. We're going to talk now about the mannose 6-phosphate signal, or the mannose 6-phosphate signal, which is going to bind to what? The mannose 6-phosphate receptor. Not surprising, right? So just to remind us the context, we talked about the endoplasmic reticulum and the trafficking to the Golgi. We talked about COP2 vesicles. We talked about the COP1 vesicles that are involved in retrograde transport and in intra-Golgi transport. And we also mentioned, and you'll be coming back to that next week when you study about endocytosis, we also mentioned the fact that intracellular vesicular transport is a uniform mechanism by which there is a GDP binding protein that anchors to the membrane, and then that recruits the assembly of the coat, and for fusion, and then you get budding of the coat from a particular organelle. And for the fusion, before fusion, we need to, or the vesicles need to undergo uncoating before they fuse with the target membrane. We mentioned the fact that there are B snares and T snares. The concept that I want you to take from that information is that vesicular traffic is a uniform mechanism. It occurs in yeast, and as to the COP2 and the COP1 vesicles and the flattering-coated vesicles, the mechanisms are similar. What is different are the particular proteins that are involved. Is that concept clear to everybody? Good. Okay, so when we're talking about sorting in the Golgi, I want you to think about what kind of mechanism, or how can we devise a mechanism by which the cell will be able, or the cell will be able to identify which proteins are supposed to go to the lysosomes and which proteins are not. Okay? Now, to give us an idea what this sorting process looks like, just to, and while you're looking at that, you can think about it in the meanwhile. So what we have here are basically two proteins. One protein is labeled with green, and the other protein is labeled with red. And as long as that they are both in the Golgi, they're overlaid with yellow color. And when one gets sorted out of the Golgi and the other one remains in the Golgi, you can see the segregation between the two colors. And this is where the sorting processes take place in the Golgi. Okay, so the tagging of lysosomal proteins. How do the lysosomal proteins, such as the lysosomal enzymes, for example, how do they get tagged so that the cell knows, or that the system knows to send them to the lysosomes? Does anybody want to speculate how one could design such a system? Yeah?
Ubiquitin?
No.
Design.
We'll talk about ubiquitin today, but not in this context. That's coming later. That's for degradation. What kind of mechanisms would we like to design? Okay, so I'll pick up from what you said in the sense that we need some sort of tagging mechanism. And ubiquitin is a tagging mechanism, but it doesn't work for this process, but for other processes which we will discuss. So the tagging of lysosomal enzymes involves the transfer of N-acetylglucosamine to mannose, and then removal of this N-acetylglucosamine, leaving a phosphorylated mannose. So what did we do here? We have our glycosylated protein. Remember, we discussed the processes of glycosylation. And now, for a particular glycosylated protein, we have an additional modification which results in the addition of a phosphate group on the mannose residue. And now this particular protein, which needs to go to the lysosome, is tagged by a phosphorylated mannose. Okay, take 1 minute to digest that. And if we say that we have a tagging mechanism, obviously we're going to have enzymes that are involved in this process of tagging, right? Because tagging doesn't happen on its own. We need an enzyme to do that. So, what's the enzyme? We have two enzymes. The first enzyme is the N-acetylglucosamine phosphotransferase, which recognizes the particular substrates that need to go to the lysosomes. Not substrates, but enzymes or proteins that need to function in the lysosome. Those are recognized by the N-acetylglucosamine phosphotransferase. And afterwards, when the N-acetylglucosamine is removed by the phosphodiesterase, there is remaining of a phosphate group on the mannose, or a phosphorylated mannose at position 6, hence mannose 6-phosphate. Okay, yeah?
So we don't take off the whole GLC and the C complex, right? We leave one phosphate on? Like, where's the phosphate
You
complex?
leave one phosphate on. I didn't want to get into the chemistry. For our purposes here, what we need to remember is that this is a two-enzyme process, the consequence of which leaves the mannose phosphorylated within the sugar tree. Okay? So obviously we have recognition mechanisms here which need to operate between two components. One is the enzyme that actually performs this process, and the second, of course, is the enzyme substrate, which is the enzyme or the protein which needs to go to be delivered to the lysosome. Yes?
You might have gone over this, but I forgot.
Yeah.
Does clathrin only go to the lysosomes, or does it go to...
Clathrin not only to the lysosomes. You will study that it is also involved in endocytosis. But for purposes of my lectures, I'm not talking about endocytosis, but you will study about that in the context when Professor Ronit Sugiaziber teaches you endocytosis. She will discuss more about the clathrin codes. But what was important for me in the context of teaching you the basic principles of protein transport, that you realize that all these transport mechanisms, they are very similar. They operate according to similar rules, but there are different proteins that is going to determine from which organelle they are going to bud off to, and to which with which organelle they're going to fuse. Yes?
So the mannose phosphate is the signaling molecule?
Yeah, I haven't finished. We're going to discuss that now for 10 minutes. And now another thing. If there may be a situation or I don't have enough time to go over a slide, but I promise you that I go over the basic concepts that you are required to remember. Okay, so let's imagine for a minute that if the enzyme, this N-acetylglucosamine phosphotransferase, if it doesn't work, what's going to happen to lysosomal enzymes? What's going to happen to them? You know what? I'll give you three options. One, they'll stay stuck in the ER. Two, they'll stay stuck in the Golgi. Three, they'll be secreted from the cell. Okay, let's vote. ER, Golgi, secreted. Okay, so I promise I'll teach you this, but I want to get into your minds. Why Golgi? Who said Golgi and is willing to explain? Yeah. Yeah. You.
Because lysosomes are formed from vessels fed off the Golgi,
Yeah.
and are targeted towards the plasma membrane
Yeah.
so that they can be exocytosed.
Yeah.
But if you don't have
You have the targeting,
lysosomes,
so what's going to happen?
Target them, then you're staying in the Golgi.
Why are they going to stay in the Golgi?
Because that's where the that's where the lysosomes come off of. That's the sensible.
Okay, who wants to argue different?
Because it doesn't have a signal to go anywhere else.
That's what's going to stay in the Golgi
No,
or go out?
go back to the ER.
That's why.
That's why one.
That's why it stays in the Golgi. Because it doesn't have a signal
Okay,
to go.
so
otherwise
remember
it
I
does not
mentioned
want
to you the other day that we have the default pathway. What does the default pathway mean? It means that if you don't have a particular signal, and assuming that you folded, that you meaning the protein, that the protein folded correctly, it will probably be secreted and not stay in the Golgi. Okay? If there is not a particular signal saying that the protein needs to stay in the Golgi, or that the protein goes elsewhere, and if there is not a folding defect, which will mean that it will get retained in the ER, then probably the protein will be secreted. Okay? Yeah.
So isn't the goal to secrete it anyway?
What?
Isn't the goal to secrete it anyway?
But this particular pathway is going to take it to the lysosomes. Not everything from the Golgi is secreted. Depending if it's going to the lysosomes or being secreted, or if it has a Golgi targeting signal. But the proteins that are destined to go to the lysosomes, them being as lysosomal proteins, they don't have a signal to stay in the Golgi. They need to incorporate the mannose 6-phosphate signal, which will direct them to the Golgi, to the lysosomes, and we'll see how that happens. Yes.
So will they stay in the cytoplasm or will they leave the cell completely?
Why do you think they'll stay in the cytoplasm? If they're in the Golgi already, how can they stay in the cytoplasm?
You said they would leave the Golgi,
They would leave the Golgi to go to the lysosomes.
but they don't have a lysosome. They don't have a lysosome.
If they don't have the lysosome signal, they will bud off from the Golgi and get secreted outside of the cell, assuming they fold it correctly.
Okay, so you don't.
You don't get cytosolic. I mean, the whole concept of what we discussed in protein trafficking is you get insertion into the ER with all of the mechanisms that we talked about, signal peptide, SRP, la 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 la. Who gets into the secretory pathway, who meaning the proteins getting into the secretory pathway, are not going to be cytoplasmic proteins. They're going to be luminal secretory proteins or luminal transmembrane and a little bit cytosolic if they're transmembrane, but they will maintain that orientation until the end of their days. We'll see maybe a little bit of a different example today, but this is the general concept. Okay? Yes?
So the answer to number one, would that be secreted or would it stay in the Golgi?
Excuse me?
So what would happen to the enzyme if these enzymes
If this
don't-
enzyme doesn't work, that's exactly what I said. It will be secreted, assuming it's folded correctly. It will be secreted because it doesn't have a Golgi retention signal,
goes-
because it's folded, and because it doesn't have a signal to go anywhere else. So it will go by the default pathway to be secreted.
So when in doubt, we always push proteins out of the cell. We don't
It's
know
not
what
a matter of doubt, it's a matter of not having a particular recognition signal. Remember, it's all about having the signals. If you don't have the signal, you get basically remote. Okay? Questions? Okay, let's get a little bit into a deeper. How can we identify a binding site to this enzyme? Binding site of a lysosomal enzyme? What do you think? How can you do that? You don't have to worry about how you actually do it in the lab. I mean, imagine, tell
Has to
me,
be
how
po-
can you figure out what's the binding site?
Has to be positive?
Pardon?
Positively charged?
Well, you were gonna, let's say you assume it's this site, you start to mutate and you see what happens, whether or not it's equal bind or not. Okay, let's continue with this. Okay, so what's gonna happen here? Let's look at the general pathway. Let's look at the general pathway and make absolute sure that all of us understand that. So the addition or the sorting to the lysosomes or addition of the phosphate tag on the mannose by these two reactions that I just mentioned takes place where? In the Golgi. So having said that, if these proteins exit the ER, what does it tell us about their folding? That folding has been completed, okay? So that comes back to what I asked you before, assuming this process doesn't take place here, the process of addition of a phosphate to the mannose of the lysosomally sorted protein doesn't take place here, probably will be manifested in secretion of this protein because it is already folded, because these processes take place beyond the ER, and because the ER is the primary site of folding in the cell. Is that clear? Kind of repeated what I said last time, but with this particular example. Yes?
So things can't get stuck in the Golgi from a misfolding problem?
It's a good question, and as you'll see in cell biology, there's never an absolute answer, but you can think about an example where there may be a need to couple with a particular protein which is present in the Golgi in order to be secreted. And if that protein is not present, or if the coupling reaction takes place in the Golgi, it might be stuck there.
And then the Golgi like explodes or something?
Well, if the Golgi explodes or not, there's not too many studies on what happens to proteins that get stuck in the Golgi. There are more studies, and we'll get back to that today when we talk about protein degradation of proteins that get stuck in the ER because of this folding, because this is the primary site of folding. In the Golgi, you get other kinds of reactions that may retain a protein or make it being transported slower, but it's not the main, the bulk. Yes?
So the signaling, the act of adding a signaling molecule is occurring in the Golgi?
The active tagging of the mannose with the phosphate takes place in the Golgi. It's a two-step reaction. It takes place in the Golgi, and the steps are what I told you before. Okay, so now what's going to happen? We as the protein is going, the protein that needs to go to the lysosome is going to the trans-Golgi network. Trans-Golgi network is where sorting of the proteins is going to take place. Sorting meaning what goes to the lysosome, what goes to be secreted, what stays in the Golgi. So there is now a clathrin-coated. You wanted to ask something? Yeah.
Assuming the protein doesn't have a signal, and so it's just
A signal for what?
signal to go anywhere. It just doesn't have it's secreted. So we probably something like that.
Probably will be secreted, assuming it's correctly folded.
So what's the difference between that and antigen? I mean, why doesn't the body recognize that as an antigen? It's just like a free.
Why should it recognize it as an antigen?
It's a free floating protein.
No. I mean, you have, bovine serum albumin is secreted. Not everything that we secrete.
Isn't its function to carry like?
We have a lot of proteins that are being secreted, like hormones and all kinds of proteins. We function because we have proteins that are also being secreted.
So, I mean, a difference in transplant.
So I'll ask me in the break because it's or I don't understand your question, but I won't go before you get a satisfying answer. Anyway, so in the trans-Golgi network, this protein that needs to go to the lysosome now is going to be budded off in a clathrin-coated vesicle. Clathrin-coated vesicle, meaning that the coat protein now is clathrin, not COP1, not COP2, but clathrin. And why will it go into the clathrin-coated vesicle? Because this clathrin-coated vesicle is also going to contain what? It's going to contain the mannose 6-phosphate receptor. It's a receptor that will recognize the phosphorylated mannose on the protein. So this will enter now this clathrin-coated vesicle. And for fusion, like we studied before, for this vesicle to fuse with the lysosome, what needs to happen to the clathrin? Needs to be removed, right? Will be removed by similar mechanisms like we discussed for the COP2, but we're not mentioning the proteins that are involved. So then this vesicle is going to fuse with the lysosome, and now what's going to happen? We have the mannose-6-phosphate receptor bound to the protein that contains mannose-6-phosphate modification. As long as it's bound in the vesicle, it's bound in the Golgi. What happens in the lysosome and why? What happens to this binding between the mannose-6-phosphate receptor and the protein? Yes? What happens? It will separate. Why will it separate? Because the lysosomes have an acidic pH. In the acidic pH, the mannose-6-phosphate receptor will not bind to mannose-6-phosphate protein. Okay? Thanks. Yeah.
Is the mannose-6-phosphate receptor continuous with the membrane of the lysosome?
The mannose-6-phosphate receptor, it's a transmembrane protein, and it basically shuttles between the lysosome and the Golgi.
It doesn't get into the lumen? Right?
No, it's a transmembrane protein. Again, the concept being that anything that enters the secretory pathway and it's a transmembrane protein, it will remain transmembrane throughout. Anything that is luminal will remain luminal throughout. So this mannose-6-phosphate receptor, it shuttles between the trans-Golgi network and the lysosomes and going back, where in the Golgi it will be able to bind mannose-6-phosphate residues on proteins. Why? Because the pH enables it. Once it shuttles to the lysosome, then this is going to separate. The mannose-6-phosphate receptor will separate from its ligand, from the mannose-6-phosphate containing protein. Okay, so dissociation. And what else happens? Another reaction happens. Another enzymatic reaction happens. And what is that reaction? That is the removal of the phosphate. And removal of the phosphate, what does that do to the protein? It activates it, exactly. So we have two steps here to ensure. Okay, so once this has been removed, what else does this ensure? It's activated, and what else can't happen?
Can't
Is it
leave.
degraded?
Can't
What?
leave.
Can't go back. Why can't it go back?
There's no way.
Because it can't recognize the mannose-6-phosphate receptor. So nobody's going to carry it back. Yes?
So what is, if you're just going to put the clathrin coat on and then just take it off right away, what's the point of it?
You were in yesterday's lecture?
Yeah.
Okay, so the point is, I'll just revive your memory. The idea is that the clathrin coat, or the COP1 coat, or the COP2 coat, the coats that are involved in generation of the vesicles, they are important for the generation of the vesicle. Once the vesicle is generated, the coat, in order for the vesicle to fuse with its target membrane, the coat needs to be removed, and underneath the coat are proteins that will recognize particular proteins on the target organelle. The coat is not required for the fusion.
But you just need it for the structure.
You need it for the budding of the vesicle, for the generation of the vesicle, and eventually for the budding. Okay, it's like a two-step process. And it actually, if you think about it, the assembly of the coat requires this GTP binding protein to be anchored in the membrane. So you have many processes that basically regulate the generation and the budding of the vesicles. It's a very, very tightly controlled process. And the more steps you have, the more it's controlled. Okay? Yes?
Can you just clarify which type of coat you use when, or is used when?
Yes, I'll collect questions and answer together. Yes?
What prevents a lysosomal enzyme from being denatured in the lysosome?
To what?
What prevents a lysosomal enzyme from getting denatured in the low pH of the lysosome?
Okay, I'll answer that too, yeah.
Is the SAR1 conserved between all coats, or is that
So the SAR one, okay, I'll answer that too, yeah.
I just want to, is the
But in a minute I'll have to stop because I forget already, yeah.
Yeah, the mature lysosomal hydrolase, that's the protein that is transported to stay in the lysosome,
Yes.
right? Do we need to know the enzyme that cleaves the M6P?
No. Yes.
So the receptors end up going back into the Golgi. Wouldn't the pH of the Golgi eventually drop, causing dissociation
Okay.
of the Golgi?
So I'll start answering because at my age I forget, so we'll start going slowly.
Is it that?
SAR1 and the homolog, or not homolog, but it's called ARF1 for the COP1 codes. They are similar but not identical. There are different proteins, but they have similar functions. It's like you have a 7 transmembrane protein. The idea of having 7 transmembranes, they're similar between 7 transmembrane proteins, but one can do a certain function and the other can do another function. So the idea of having a GTP binding protein that binds to the vesicle and recruits the coat proteins. Coat proteins meaning COP1, COP2, clathrin, and possibly others is similar. The idea that fusion occurs only after removal of the coat is similar for all of the coats that we discussed. The proteins are different, but their function in terms of what they are doing in this process of transport is similar. Okay? Yeah. What else? All other questions didn't I answer? Yeah?
Just one each type of code.
Oh, okay. I'll summarize that. Yeah.
Why is the lysosomal enzyme not degraded in
Okay, so the codes that I discussed with you are the COP2 codes, which are important for the transport from the ER to the Golgi. The COP1 code is important for retrograde transport from the Golgi to the ER and also for intra-Golgi transport. Golgi transport from one segment of the Golgi to another, as well as retrograde transport to the ER. Clathrin codes are going to be important for transport from the Golgi to the lysosomes. And you'll also study about clathrin codes in context of endocytosis when you study endocytosis, I believe, next week. There was another question that was concerning the fact that if an acidic vesicle buds off from the lysosome and goes to the Golgi, will that make the Golgi more acidic? So it doesn't make the Golgi more acidic because of size considerations and volume considerations. It's not enough to make it acidic, plus there's all kinds of pumps that take care of that. Yes?
Yeah.
Okay, yes?
So if it's being transferred from, let's say, the cis part of the Golgi to the trans, does that mean that it has a COP1?
Yes, if it were to be transferred from here for here, this will go by COP1 codes and onwards by clathrin. Yes?
When the phosphate is removed, is the mannose also removed, or is the mannose a permanent part
No,
of
the mannose will not be removed, the phosphate will be removed.
Okay.
So the protein being in the lysosome, what is characteristic of the lysosomal enzymes, and that might answer your question, is that their maximal activity is in low pH. So, and if you think about it, it's a beautiful mechanism. We'll discuss it a little bit when we talk about the proper degradation lecture. It's a very elegant mechanism to avoid activity of, let's say something happens to our lysosomes and the membranes and the lysosomes for some reason get disintegrated. What will happen if these lysosomal enzymes, which are chop enzymes, get in the cytosol? Will something happen?
Yes. Lysosomal storage.
Probably not, because they won't be active in the pH of the cytosol. Okay, that clear? Because their maximal activity is at low pH. And the low pH is characteristic specifically of the lysosomes. So if, and the lysosomes, again, they are surrounded by a membrane. So this low pH is within a contained environment. Okay, so if something happens to the lysosomes, and let's say the lysosomal enzymes get dispersed, then they won't be active. Now think about it even from another point of view. What's the advantage of having the mannose 6-phosphate modification performed in the Golgi. What will that avoid? What will that prevent? Prevent is the word.
Activation.
Yeah. Activation. So remember, we're talking about the secretory component. We just made all of the proteins that we wanted to make. We finally made it out to the Golgi. We don't want to be subjected to the chance that proteins within the Golgi may be, who knows what, degraded even a little bit by these enzymes. Not maximal, because of course it's not the maximal pH, but we want to be absolutely sure that they are inactive at this point. Yeah?
What about transmembrane proteins in general? Do they stay on the membrane while they bud? Or
Yes, yes, the budding is within, you can see it here. The mannose-6-phosphate receptor in the green, it's within the membrane, it buds in the membrane, it
But
stays there all the time.
transmembrane proteins that are formed in the rough ER to be not just for lysosomal, just in general.
Yeah. Every receptor that you have on the cell surface, EGF receptor, VEGF receptor, endothelin receptor, transferrin receptor, any kind of receptor is formed here and makes it all the way to the plasma membrane by the mechanisms that we discussed.
So when it's going from like the rough ER to the Golgi, it's still
It
on the
will
membrane.
go in the vesicles and its orientation will remain such that the luminal part will become extracellular, the transmembrane will always stay transmembrane, and cytoplasmic portion will also always stay cytoplasmic. Questions? Everybody's happy with this? Yeah, good. Okay, so removal of the phosphate, the dissociation at the acidic pH, and the recycling via the clathrin coats. Okay, diseases. You want to know about diseases. So, eye-cell disease. Did you ever hear about eye-cell disease?
No.
Yeah, great. Okay. So, eye-cell disease, it's also called mucolipidosis. Somebody asked me about lysosomal storage diseases, I think, at one of the lectures, and I said that I will come back to this. So, eye-cell disease, extremely rare, 1 in 350,000. And probably a lot of these genetic diseases that we study about and that they give us some sort of hint also on basic cell biology processes, a lot of them, once the genes have been identified and genetic screening becomes more and more available, they will, of course, become lower and lower in the population. But at this time, so this is identified as the eye-cell disease. And what's the problem in eye-cell disease? It's deficient activity of which enzyme? The N-acetyl-glucosamine 1-phosphotransferase. Remember what this one does? It's the first step that adds the modification before the removal of the N-acetyl-glucosamine and the remaining of the phosphate on the mannose. But the defect in this enzyme would imply that there is no mannose-6-phosphate tagging, right? Yes?
Mm.
Okay. So actually this is an animal model of this human disease, and it was found in domestic short-haired cats. That basically, what did they find? They found that lysosomal enzymes from affected kittens compared to their normal adult control cats. What did they have? They had high serum levels, a lot higher, like 10 to almost 100 times the normal one, but low in the cultures, right? And in the cultures, where am I? Okay. And why were there inclusion bodies in these cultures, in the lysosomes? Why were there inclusion bodies? What didn't happen?
The lysosome didn't hydrolyze them.
The protein degradation didn't take place in the lysosome, so substrates accumulate in the lysosome, and therefore one gets inclusion bodies. Is that clear? Yeah? Okay, so basically we find in these cats, and we'll see that also in the patients, we can find higher levels in the serum of lysosomal enzymes. The reason is because they are not targeted to the lysosomes. The enzymes are not targeted to the lysosomes. Yes?
So would they be active in the serum or not at all?
Ah, it's a great question. I think think.
'Cause they don't
It's a
have a
matter of
phosphate.
thinking. I don't, I'm not sure. I'm not sure. I'm not sure, but it's a great question. If you find a good answer, then they didn't find anything that alluded to that, but it's a good question. Yeah,
Does that mean that they're active before they get to the Golgi?
sorry?
Does that mean that they're active before they get to the Golgi?
You know, it's a question, theoretically maybe yes, but we're talking pH again, so they can be active in the higher pH, that's one thing. And the second thing, for every protein, there is a different kinetics, different folding kinetics and a different transport kinetics. And I don't know what the transport kinetics are for these proteins, but it might be that it's extremely fast so that they are very fastly modified. But again, it's speculation, but it's very relevant. I mean, it's relevant. Yeah? More? So if you look at these cats, I'm not a cat grower, but apparently they have more I don't know how you say it in English, pachus, like flat, more a flatter face, and they have these inclusion bodies inside their cells. So obviously, to have an animal model for a disease, what is that good for? Study medications, study mechanisms, try new drugs, whatever. It's good for a lot of things. And apparently this is the only animal model that there is for this disease. And if you look at patients with this disease, you also see this flat appearance. And obviously they are not, they don't live long because it's a very, very severe defect. Okay, so now we're taking deep breath and we're turning to the degradation lecture, but you can obviously see how it's connected to the end of the previous lecture. So the lysosomes, they have very many different enzymes there that can hydrolyze proteins, RNA, DNA, polysaccharides, and lipids. And mutations in genes encoding for these enzymes can be responsible for different genetic diseases, which are classified together as lysosomal storage diseases. An example of that is Gaucher. Did you study of any lysosomal enzyme?
Yeah.
Gaucher? You talked about
Gaucher?
How do you call it?
Gaucher. Oh, Gaucher.
Come on.
Gaucher. Do you know what the treatment now is for Gaucher? Enzyme replacement therapy, right? You can actually inject the enzyme, and these patients are extremely happy to get the.
Can't do that for mucolipidosis.
For mucolipidosis, well.
How do you get the enzyme to the right place? No?
Yeah, well, that's biotechnology kind of experiments to get the right targets, to get the right. Anyway, so obviously the degree of severity of a disease, if it's a lysosomal storage disease, will be based on whatever the substrate is, and also, well, for Gaucher there is a treatment, and then there can also be maybe one can devise other kind of strategies to ameliorate the symptoms of disease. Can you think about a trivial one? About a trivial kind of approach? So if we know what a substrate is, for example, for an enzyme, then we could possibly devise a way to get less of the substrate, for example, to accumulate less of the substrate, which will also have so this is also a concept. Okay, and eye cell disease we discussed. So the enzyme replacement therapy we talked about. And now when we talk about the protein degradation, again, I think that by this point of our studies, we can realize that unlike the Polish mother, having more is not always better. Sometimes more is not better. And we have to realize that when we're talking about homeostasis, we're talking about a flexible kind of situation and not a fixed state. So we have a dynamic process where we have protein synthesis, like we studied, and we also need to take care of protein degradation. And again, the level of the proteins at steady state are going to be the size sum of the synthesis and processes of degradation. And again, the total levels may appear constant, but the cell can perform changes as required. And when we're talking about protein degradation, obviously there's the extracellular pathways, which have to do with the breakdown of proteins in the intestine, and we're not going to be talking about that at all today. And there are intracellular pathways. There are going to be lysosomal degradation and degradation in the cytosol. And we discussed, but this is not something that we're going to talk about now, it's just to give you everything in the general context again. We talked about the cleavage of the signal peptide from proteins in the secretory pathway, but so that's also some mode of degradation, but we're not going to be talking about that now. So we're going to concentrate now on intracellular protein degradation in lysosomes and in the cytosol. Okay, how are we with time? Do you want to go straight like until 3:15, or do you want to have a break? No problem. So we have a break what time? So I plan my moves. Ma?
Whatever time.
Already?
No, 2:30.
2:30? Okay, let's finish this concept and then we'll go from there. Okay, so when we're talking about intracellular proteolysis, basically we're exchanging about 5% of our proteins daily. How come? Why should we exchange? Because basically this is going to enable us to control the activity of short-lived processes such as signaling, such as cell cycle, like we'll be talking about on Sunday with me. Yeah. And also we need, remember, we still have a problem we need to deal with. What are we going to do with all the improperly folded proteins that we generated in the ER, especially if they're mutated or just unfolded properly? How are we going to get rid of those proteins? And obviously we need to maintain the cell functions at steady state. So when we talk about protein stability, the proteins, different proteins have different half-lives, different extent of stability. For example, hemoglobin, its stability is around 120 days. Certain cell cycle proteins are very unstable and unstable or unstable? Unstable. Unstable. And like in the minutes range, and cell surface receptors, we have some long-lived receptors, we have some short-lived receptors, and for lens proteins, some of them can live as long as the organism. So obviously, this information, what's the challenge in? What's the challenge in this information?
Maintaining balance.
Sorry?
Maintaining a balance.
Maintain the balance is a challenge, but what is the molecular challenge? Yeah.
For degrading them if they all have different half-lives.
How do you know which protein should be degraded and which protein shouldn't be degraded? How can we differentiate between a cell cycle associated protein which half-life is in the minutes range from hemoglobin, which half-life is in the days range, okay? And that's going to be extremely fundamental in controlling the processes in the cell. Also, sometimes we have a complex of proteins, of two proteins, where one subunit is very stable, the other one is extremely unstable. How do we differentiate between them in terms of managing to degrade? So when we want to design systems for degradation, we have to make sure that degradation takes place at the right time, and we have to have a specificity of the degrading machinery, okay? So these requirements are going to dictate, again, a recognition mechanism, which I discussed. And again, we're going to need a specific, so one is going to be to recognize, the second one, we need the executor. Okay? We need somebody to execute the degradation. So two aspects to that. So we talked about the lysosomes, and I don't think that I need to expand too much on that. But we talked about the acidic pH. That is important. That is a basic concept that you have to remember. Also, the lysosomes can also degrade worn out organelles. And these lysosomes will handle products of receptor-mediated endocytosis. You haven't studied endocytosis yet, but when you will study that the degradation eventually is going to take place in the lysosomes. Now, a very important landmark in the study of protein degradation was the scientist called Rudolf Schonheimer. A little bit of history never hurts. And he was a German biochemist, and he developed the technique of radioactive tagging of molecules, which will enable to tell us how stable a molecule is. Because if we can tag a protein with radioactive labeling, then we can follow how stable that protein is by virtue of the fact that it is labeled by a radioactive label. So this particular scientist moved in 1933, from Germany, when Europe was extremely stormy, to Columbia University, and there he came up with this paper on studies on protein metabolism. Okay, so when we're talking about protein stability, obviously it's the amount of protein at a given point, and again we said that this can very much affect the cellular activity. So now I'm going to give you going to ask you a question, and we're going to try to answer that, and then I'll give you a break. So I'm going to tell you the following fact, and the fact is that the lysosomes degrade mainly extracellular components that were internalized by the cell. Okay? So I want to distinguish again and to make myself clear. We were in the Golgi, and we transported the enzymes involved in degradation, we transported them to the lysosomes from the Golgi, right? So we have the machinery already in the lysosomes. And now I'm telling you that this machinery and that the lysosomes degrade mainly substrates that are internalized from outside of the cell. Not intracellular proteins, but proteins that are internalized from outside, from the extracellular milieu of the cell. How can you design an experiment that will prove this point? Yeah?
Tag it and then
Tag what?
Tag the protein extracellularly.
Okay.
Or so it's built and it goes
Yeah.
extracellular.
Yeah.
And then see when it goes back in.
Okay, but how are you going to show? Yeah, okay.
Could you tag the plasma membrane to see if it does the endocytosis and then fuses with the lysosome?
So what you are both saying is you're giving me examples of how can I show that extracellular proteins are degraded by the lysosomes. But that wasn't my question. I told you that the lysosomes degrade mainly extracellular proteins. You're not. I want you to exclude the fact that they are degrading intracellular proteins, not that they are degrading extracellular. That is, that's good. They degrade extracellular. You showed me that they degrade extracellular proteins. I want you to design an experiment that will tell me that they don't degrade intracellular proteins. Okay? Yes?
Can you have the cells grow using a heavier isotope of some,
Something, never mind, yeah.
and then you move it into like a normal media of extracellular media, and then you see what's degraded, and if you have a heavier isotope of the normal.
So you're almost there. The idea is, again, and that's why I told you about the labeling of the isotopes, the idea is to have two populations of cells, which are going to be labeled with two different labels. The cells that we're going to choose are cells that, what do they know to do? To phagocytose, okay? To internalize all kinds of extracellular components. Now we're going to have two populations of these cells. Each one of them will be labeled with a different isotope. Okay? So we're going to leave one of these cells intact. So one cell is intact and labeled with a certain isotope. The other cell that is labeled with the other isotope, we're going to lyse it. We're going to get rid of the membranes. They're just going to have free-floating proteins, but they're going to be labeled, right, with the isotopes. Now, what's going to happen if we mix the intact cell with the mixture of the proteins from the other cell that was also labeled, but with a different label? What are we going to see in those cells? based on what I want you to prove.
Or the heavier one.
So we're going to see in the lysosomes the proteins which are labeled with the isotope from the outside, but the same proteins from the inside the cell will not be in the lysosome. Okay? Yeah.
That you've demonstrated that by this experiment that a cell specializing in phagocytosis would lyse more external than internal components. But can you use that same experiment to characterize a cell where phagocytosis is not its primary function?
You could theoretically do that.
It
would
makes logical
take
sense
less, but
that.
you could do that.
Okay.
Yeah.
And it would still be
It's
primarily
same idea. Yeah, I'll show you the paper in a minute. Yeah.
Are you saying mix the cell populations, one with a cell that has a membrane intact, one that has like no membrane because you lysed it?
Yeah.
And which proteins are you using? I'm just
You're going to
confused.
label the entire protein population of the cell, and then you're going to find out which, if they were incorporated into the lysosomes, the one from the outside.
Doesn't that just
Or you could extend it to one kind of protein. So this was the actual experiment that was done. It was performed in 1978 by Brian Poole. Was anybody born then? Well, I was already pretty old, but Brian Poole. What did he do? He labeled metabolically endogenous proteins in living macrophages with leucine labeled tag, and he fed them with broken macrophages that were labeled with the C14 leucine prior to their disruption. So we have two kinds of label now. So they assumed, apparently correctly, that the dead macrophages, debris, and proteins will be phagocytized by the live macrophages and targeted for lysosomes for degradation. Now, what they found is, when they inhibited this degradation, that the drugs inhibited specifically, the drugs that inhibit lysosomal degradation. What kind of drugs do you think can inhibit lysosomal degradation? What would they do? Increase the pH of degradation. If there was a drug that could selectively increase the pH of the lysosome, so those drugs inhibited specifically the degradation of the extracellular proteins, but not the intracellular proteins.
Why?
What was their conclusion? Their conclusion was that exogenous proteins will be broken down in the lysosomes, while endogenous proteins will be broken down, 1978, the PNAS paper, wherever it is that endogenous proteins are broken down during protein turnover. 1978.
What does that mean?
Yeah, whatever it is.
They didn't know.
Oh, okay.
Okay, that's why we have another hour for this lecture, because I want to tell you wherever it is. You don't believe for 1 second that I'm going to leave you in 1978. We have to tell the history, what happened, because we should appreciate how science evolves and what is the process of thinking of the people who are conducting it. So again, we're going to need to design an intracellular cytosolic machinery that is going to degrade intracellular proteins. And we're going to think about free versus structure complex and recognition mechanisms. So I have 5 more minutes until your break? Or you want to nap? Or what?
Let's just.
What? What do you want? Tell me what you want. Okay, so take break now. It will be easier for me to continue and come back at 22. Would that be all right? So 15 minutes. 20 to yeah, 15 minutes.
An hour.
Yeah, sorry.
These are just like the I can stuff. But they're extracellular cells.
I didn't get it. But it's okay, it doesn't matter, whatever. I'll show you how to send it actually. I didn't send it this way. Here, you do it like this. It's over, it's over. Now I know it's down here. Over here.
Oh, yeah,
So your assemblance cellular not all cellular products are
Can't do this.
No, because if you have one cell, then all of the proteins that you have, none of them are going to be like.
Yeah, no, I get it.
the idea is correct. I don't know if I don't know if it should be. I think that's
Dude, install this app. Yo, that's a bomb ass app. Got it? That's what I was looking at you for. What's it called again?
No, we're saying suppose we get a minus 6 phosphate. It wasn't modified, it's supposed to go to the lysosome, didn't go to the lysosome, it was secreted, so the immune system should recognize it. What I'm saying is, I don't know how to, basically, the immune system is not supposed to recognize antigens which they give tolerance towards.
Dude, where was this shot?
They don't, I don't know, is what is the mechanism of building tolerance towards cellular proteins like that? Yeah,
I just do very good.
I don't know.
I'm like obsessed. I'm literally gonna do that again today. Like before people class is like 5%.
No, it's not easy to touch and feel very good.
I know, isn't that crazy? Like, think about how tired you are, and then I don't know
The A is O to touch N, the N is
Don't think that the level in the serum is zero.
Yeah, I think honestly, I'd like to see
Might be that it's really bigger than that.
because if I'm gonna do something, I want to.
But the idea is correct, that you have this targeting and if this system is outside of the cell. You know, there's, for example, along that line, there are situations where the hormone.
Do you really? Me and Lior were just really crazy about it. This is a stupid question. We're just wondering, like, you know, like, continuous
Maybe 11. These are the hormones that you get.
Is there like a separate mechanism? Or do they just show up from cell division? Is that worth asking?
I don't think she'll know. Maybe she'll do this.
Well, like, people like really want to know.
Yeah, that's interesting. I don't know. I wonder if she'll even
It's kind of a meta question.
Yeah, like once it's on your computer, it's because it just takes forever. Everyone just like install.
It might be.
You can't just get it in the App Store? So they were all in the App Store?
Just give me your computer. We're hacking the Chinese.
My hormone, based on maybe or activity of other enzymes inside the cell. And when you make a recombinant hormone, you have the entire population. It's not a hormone protein or it's not exactly what the hormone is like. So there are able to develop antibodies to hormones that they're treated with because of this situation. But they're not. Obviously, the pharma companies would, collapse it definitely with the mainstream. But it's not unheard of. It's not unheard of. And in terms of your what happens?
It's a stupid question.
Cells are they recognized by immune system?
There's no answer. It's like asking how did life start?
Yeah, it's like that. Take it off, and then once it's on your computer, you can install it.
Well, there might be leakage in the sense that even healthy cells, if they undergo apoptosis,
something makes sense to see classified usage in general.
Right. For example, or I don't know, for the immune system.
Didn't write my name again. You didn't write it yet? Right in your class.
Have exposure to antigens
No, I read it.
afterwards, don't get exposed, or vice versa. So there's no like one definite answer, but it's definitely an issue, what you raised. And again, it can also actually portray on treatments that have to do with
Clarification question?
Sorry?
Clarification question?
No, there's just a bunch of slides from last lecture that weren't covered, so it's actually
Oh, I think they were, you know what? Oh, the slides, which ones, about the what?
like treatment approaches for lysosomal storage diseases and stuff.
Ask her if you have to know it.
That's what I'm doing.
Post it on Facebook.
Ben, do you want me to clarify a concept?
Yes. The classic.
For the extracurriculars, when you'll start
But I clarified during class, not up here. Yeah, I'm not part of the like clarifying concept.
say, well, whatever. Okay, so
It's the second day I've gone up to ask. Hi.
Yes.
I was just wondering, in the last, like when you originally posted the lecture, there were these slides.
Which one are we talking about?
Protein-specific secretion,
yeah,
like
yeah,
treatment
yeah.
approaches.
Yeah.
And then, well, this was.
Well, this,
moved
you know, it's
earlier.
all self-explanatory. This one I showed you.
Yeah, yeah.
It's just like the ones.
What's the fourth one called? The
Okay.
Pharyngeal? What's it called? Trochlear?
Also, just for like in terms of studying for the quiz or anything, the slides the slides are very like good and like lots of words and details. Like those should be fine in terms of conception.
Normally, where I think that there's basic concepts,
Yeah.
I pause and I say,
Yeah, no, I know. Like
Like I
I've
mapped out like all the nerves, like
yeah.
from the
Oh,
trigeminal.
okay.
That's
Sorry.
no, it's good. Like, it's good. Like it solidifies.
Where does it go? Like, you know, like
That's
the
the two
three different parts of the
nice.
trigeminal.
Yeah.
Just go like.
Your former
and then keep going up
ancestors.
and encourage the other.
Yeah.
What do you
Oh, yeah,
like?
just use it
Like
as well.
60% of the first one?
Yeah.
Good stuff.
Good classes.
And I'll share them on
A lot of
Facebook or something like that.
slides. Yeah. It's not because of the lecture.
Yeah. Wait, what's the word?
Yeah.
What's
I
that?
don't know what happens in the States.
Oh, yeah. Yeah, where is it?
Yeah, I had for physics. I'm from Canada, Toronto.
Copy
and
through
I had
computer.
to attend the person lecture hall.
And,
Class of 800. 60 people showed up to
It's
each
there's only
person's
two
class.
files on the
In the summer course, one's lecture in front of me.
Yeah.
Like there might have been one person
Right.
as a German professor, thickest German ever, and people like didn't learn from him, and they just didn't go. But when 60 out of 8 didn't show up, there's probably a problem. Yeah.
I know that in the program, they, certain point, I don't know if it's this year or not, they introduced for self-learning topics.
Yeah.
Question is, you know, the same or not?
But I prefer, like, I find that coming here is kind of like, there's something else to do, I prefer, like, sort of hearing it from someone else telling you, like, how it works and how it works better than if you just sat and looked at it a bunch of times, you say, okay, this is this, but when someone tells you it,
The question is, if the person who looks at it says, this is this, within 2 months,
Yeah.
after he takes the exam,
We'll still
ask him
remember
the
it.
question, will you remember the concepts, or will you just here for the exam, you know, right?
yeah,
Go to touch,
yeah.
to stretch and abduction.
We're debating a lot this question of how is teaching going.
No, the fifth one.
Yeah.
Trigeminal.
Still come, I think it's useful.
What's up? Do we need to know it, Adam?
Yes.
Yes? Forget about it.
But it's just like review of what we've done.
Okay.
So it's not new. Except exocytosis, you need to learn.
Very green vegetables.
Thank you.
Yeah, you're welcome.
Yeah, we can do that one also, but.
Do you like my chart?
I mean, everything, what do you mean?
Do you like the chart?
Yeah, it's beautiful. Because you what?
Because I made it.
I don't, I'm sending it back to you. I'm just.
You do you. You do you.
What chair? I want a chair.
See which what is the last name?
You don't like it? You don't have MacBook? What do you mean, it's right here? You're not motivated.
But I'm in a programming course.
What course?
Yeah, that might not make sense.
Oh, do you want the program?
No, I'll get it.
Above? Abducens nerve?
Yes? So we're talking now about cellular systems that are involved in degradation, and we're going to talk about cytosolic degradation. So there were cytosolic proteolytic enzymes, for example, caspases and calpains, and you're going to learn about the caspases quite a bit when you're talking about apoptosis in your class about apoptosis. Both enzymes are cysteine proteases. What does that mean? It means that they have a cysteine residue in their active site. Now, since these enzymes are active in the cytosol, what would that imply? It would imply that they have to reside as inactive proenzymes. How come?
They're just all the enzymes.
Because you don't want them to be active all the time. You want them to be active only upon a certain signal. The other thing is they have a defined cleavage site. Why is that important? Unlike the lysosomal enzymes that chop, 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 chop. Why would that be important? Because then they would cleave everything. If they have defined cleavage sites, it would mean that they have a limited set of substrates and they also produce limited amounts of fragments. And what would that mean when I say limited amount of fragments? Take it further. These fragments that they produce may also have functions. Now there are differences between these proteases. You don't have to remember these differences. You will be studying a lot about the caspases and the calpains. I'm just telling you about that as well. So some substrates could be cleaved by both enzymes, and that would again introduce another mode of complexity when is it cleaved by a certain enzyme and when by caspases and when by coupling. And now we're going to move to the proteasome until the end of this lecture. What are the proteasomes? The proteasomes are large complexes of proteolytic enzymes, many enzymes. They are present in the cytosol and also in the nucleus. Just for a number-wise, not that you have to remember it, but to give you a feeling, there's several tens of thousands proteasomes in each cell. And the proteasomes, they degrade tagged proteins. So what is the tag? What's the tag? The tag is the ubiquitin, and what is going to be the recognition mechanism, and we're going to talk about that. And when we talk about the proteasome, so basically it's like a barrel, and the barrel has a cap on both sides, and degradation takes place inside here. So it's well protected from the cytosolic or from the nuclear environment. So the tag is ubiquitin, and ubiquitin was originally isolated from the thymus in 1975, and at that time it was thought to be a hormone. Later on it was found in all tissues, and then it was granted the name ubiquitin. Subsequently, it was also found to be part of a branched protein, 1977. And later on, intracellular protein degradation was found to require energy, or ATP. And this was found also in a cell-free system that was designed specifically to study degradation. And in this system, using this system, they identified ubiquitin. And what is ubiquitin? It's a heat-stable protein, and this is how it was identified. And the paper that identified was by Chikanover et al., 1978. And based on these findings, it was suggested that addition or ligation of ubiquitin to a protein marks it for degradation by a protease. That's what was said in that particular paper in 1980. That specifically recognizes ubiquitinated protein. Now notice, historically, we're talking 1980, and it was the paper in 1978 that said that we're looking for where cytosolic proteins are being degraded, and this was now addressed in 1980. What is ubiquitination? So this pathway is initiated by ATP-dependent activation of the ubiquitin by an enzyme called E1, and then one gets additional reactions by which eventually ubiquitin is going to be passed to an enzyme called E2, and together with the E3 ubiquitin ligase, ubiquitin will be added onto the substrate protein. Okay? And often after addition of one ubiquitin, we're going to have addition of additional ubiquitins on the substrate. Now, most proteins which are degraded in the cytosol are going to be delivered to the proteasomes, and the proteasomes are going to degrade proteins that were marked for degradation by ubiquitin binding. And to show you in the scheme what I just told you before, so if this is ubiquitin, this little red dot here, it will be activated by the E1 enzyme, passed on to the E2 enzyme, and then together, or by the E3 enzyme, it will be added to the substrate protein. Subsequently, ubiquitin will be added to another ubiquitin, will be added to another ubiquitin, until a polyubiquitin chain is formed. And that polyubiquitin chain is recognized by the proteasome, which will degrade the protein into very small peptides. And this is, if you want to imagine how it happens, inside the barrel of the proteasome. Yes?
Just to confirm, proteasomes degrade intracellular proteins?
Yes. Yes?
Sorry, I'm a little bit confused.
Yeah.
Where is the actual protein that is being ubiquitinated? E1 is assigning the ubiquitin?
This is the protein that is going to be ubiquitinated.
So that happens after
And the
E3.
one that is going to add it to it is the E3
Got it.
enzyme.
Thanks.
Okay, yes?
So if a proteasome needs to be degraded, is it degraded by another proteasome?
I don't know to tell you about the metabolism of the proteasome, who takes care of the proteasome. I don't know. I think the components are pretty stable there for the proteasome. Think about it also when the cell divides, so more proteasomes needs to be generated as well.
Yeah.
Very complicated, isn't it? It's a wonder it works, actually. Anyway, there were all kinds of papers talking about the minimal length of the ubiquitin chain that is required for that, but we're not getting into that. Now I'm going to take a minute because this is the heart of the matter. Okay, so we're going to take a minute here and make sure that we understand what's going on here. So this gives us an outlay of the ubiquitin system and how the principles of how it may operate. So if we're talking about, okay, let's start actually from the bottom here. These are going to be different substrates that are going to be subjected to ubiquitination, subjected to the addition of ubiquitin residues or ubiquitin tagging by the ubiquitin system. So for a functional ubiquitin system to operate, we need three types of enzymes. We have the E1 enzyme, which activates ubiquitin. So E1, what's the substrate for E1? Ubiquitin. It has only one kind of substrate. So what's going to happen if our E1 is not functioning?
No tagging.
Not good. Not good, right? Not advisable. Then we won't have any activation of ubiquitin. Basically, we won't have ubiquitin tagging if this enzyme is not working. Is that clear? It's the top of the pyramid. You have many molecules of this enzyme, but one type of this enzyme. E2 is going to transfer the ubiquitin, or together with the E3, these are both ubiquitin ligases. Ligases because they take the ubiquitin and add it onto what? Onto the substrate molecules. Now this picture here, or this outlet here, is going to demonstrate to us the complexity of the ubiquitin system. And what do I mean by complexity? And it gives us different kinds of scenarios on how this system might be regulating protein degradation. For example, and you don't have to remember any of the names here, but it's for you to understand the complexity of the system. So E3 is going to add ubiquitin residues to a substrate protein. Okay, this is one E3 that can add a ubiquitin residues to this substrate protein. This E3, for example, will add ubiquitin residues to this substrate protein, but it can also ubiquitinate other substrate proteins. Okay? There are situations where E3s, where a certain E3 can have one substrate, and there are cases where a certain E3 can have more than one substrate. Yes?
So ubiquitin could go with any protein, but E3 is specific to certain kinds of proteins.
Ubiquitin can be attached to any protein, providing, and we'll come to that, the protein has the right sequence, because not every protein is ubiquitinated. So there has to be the signal that is going to activate the E3 or change the substrate so that the E3 will recognize it. But, say again what you want?
You guys
I have answered? Okay.
Wait, question.
Yeah.
What's the difference between E2 and E3?
So E2 and E3 is basically the final enzyme, and there's also E4s now, but it's like a pyramid. This E3 is going to be the one that will eventually attach the ubiquitin to the substrate. Sometimes certain E3s need the E2s as well. Sometimes E2s can do it directly. The general concept is that the E3 ligates, or E3 adds the ubiquitin to the substrate protein. So one E3 can have more than one substrate, and there can be situations where an E3 has only one substrate. And we'll see different examples here. We can have, for example, another situation where two different kind of E3s will recognize the same substrate. And we can imagine scenarios where this regulation, where this kind of outline can increase complexity, right? Depending on signals which emerge, for example, to this protein so that it is recognized by a certain E3 ligase and not another E3 ligase, depending on the particular physiological situation that we are coping with. Questions? Yes?
What's activating the E1?
It's a great question. E1 is kind of steady state, which needs to be all the time, always a little bit active, so ubiquitin molecules basically will be more active. What's going to determine eventually the process is the activity of the E3 enzymes.
So if the proteins have the sequence the entire time, what determines if it's being tagged or not?
Okay, so let me give you a scenario. What's going to determine if they are being tagged or not by ubiquitin is either, well, probably when I'm saying either or, and there's other options as well. It can be a situation where, for example, there is a signal, an intracellular signal, that will activate an E3 ligase. And once this E3 ligase is activated, it could be activated by phosphorylation, it could be activated by maybe some sort of cleavage, it could be activated maybe by binding to another subunit. When we're saying activation, I'm talking in general terms. Anything of that sort could be activating this E3 ligase to recognize the substrate. Another option is, depending on which E3 ligases and which proteins we're talking about, that the activation will be on the protein. So let's say something happens to this protein, and only now the E3 ligase will recognize it. So it's bidirectional.
But if it can recognize multiple proteins, can it just degrade the wrong protein?
But it will recognize the multiple proteins based on particular physiological scenarios. Okay? It will recognize only if, let's say, for example, I'm not saying it about a particular protein, but it might be the case that when this protein is phosphorylated, only then the E3 recognizes it. And it's phosphorylated only if something else happened before. So that's the physiological context of how this or when this happens. Yes?
So the conformation of the substrate,
yes,
that only exists when the protein's already been used? It's like not necessary anymore?
Not necessarily, well,
or those conformations
okay,
only
not
exist
necessary is one way to put it, but it might be, for example, that one of these proteins that will be removed is an inhibitory protein to make another protein active.
Okay.
I mean, let your imagination run wild. Anything you can imagine can happen in this context where these are the basic players and variability is extreme. Complexity, not variability. Complexity is extreme. So anything in that respect can, and we have to keep an open mind as to the relationship between the E3 and the substrates, and how many substrates can go per E3, and how many E3s can recognize one substrate, two substrates, three substrates. And sometimes when you have redundancy, it's good, because if there is some sort of disease, let's say, in one, the system can still overcome. Yes?
Does the amount of polyubiquitination that's required depend on, like, I see mitotic regulators and beta-catenin are over on this one ubiquitin side of the screen. Does that mean that they're easier to degrade because you don't want to have them build up and cause cancer?
There is no rule. Every scenario is based on the proteins involved and the physiological stimuli that will drive it to take place. So, a mutation in E1, is it going to be viable? No, it won't be viable. Mutation in E2, depending which E2, and E3, also depending which E3, right? Because it might be a very important E3 that you can't live without, but most probably, and there are examples, and we will see examples, where there are mutation in E3 and how that leads to a disease. Okay? So keep this in mind. This is the basic scheme, and I purposely took more time to to talk about it because this is the fundamentals of the system. And from now on, basically what I'm going to be telling you are more examples of these. So everybody understands this E1, E2, E3, ubiquitination, degradation, protasome? Yeah, nobody fainted from that list? Good. Okay. So who determines the specificity? Which enzyme determines the specificity? E3, right? Yeah? E3. Okay. This I'm just going to skip. It's I'm just gonna give it. I gave it because I want to give credit to Warshawski, who at a certain point thought that the lifespan also depends on the N-terminus of a protein. Eventually, this rule was not so tight, and it's not something that you have to remember. But we want to talk about the Lasker Award, who got for Chekanover. He's from Haifa. Hershko, who was his mentor. By the way, he was a medical student, so you still have a chance. And Warshawski. And they got the Lasker Award for the discovery and recognition of the broad significance of the ubiquitin system of regulated protein degradation, a fundamental process that influences vital cellular events, including cell cycle, malignant transformation, and responses to inflammation and immunity. Now, see what happened to Warshawski. He was replaced by Irvin Ross, and later on, in 2004, they all got Chekanover, Hershko, and Irvin Ross got the Nobel Prize in chemistry for the discovery of ubiquitin-mediated protein degradation. They say that 50% of those who get the Lasker Award gets the Nobel, so here it's almost like that. Okay, so the paper that warranted them this Nobel Award was the 1979 paper of Hershko, Chekanover, and Irvin Ross, the resolution of the ATP-dependent proteolytic system from reticulocytes, and that's when they characterized the protein ubiquitin. Okay, so which kind of cellular proteins are degraded via the ubiquitin protasome system? Those are proteins that are associated with cell cycle. We'll see that on Sunday. Transcription factors, certain cells, surface receptors, and in most cases substrates are polyubiquitinated. However, there are examples of the addition of one ubiquitin or no ubiquitin at all. Now the proteasome is also an emerging therapeutic target in cancer. And this is actually, to my opinion, a very exciting finding. And there is actually a drug called bortezomib, which is used for a multiple myeloma. And this was a study in 2005. And this is what was concluded at that time. That was a large study, showed that bortezomib, or it's called Velcade, the first drug of its kind. So the bortezomib inhibits the proteasome, is twice as good as treating an incurable cancer as a conventional medication. And we're talking about multiple myeloma. So to other cancer researchers, the findings represent the first wave of pendulum from a molecular understanding of how drugs work against disease. And but I think that the beauty is really in this paragraph here, that the disease responses for this clinical trial and the survival advantage were so striking that the trial arm was ended earlier, and people in the control arm were actually switched to bortezomib, and the FDA, and you all can appreciate that the FDA doesn't have a light hand on introducing new drug, extended its approval for use as a last resource to include myeloma patients of a prior, who have received at least one prior treatment. So if you ever study about clinical trials, I think it's not the common case that during a clinical trial, the differences between the control arm and the treated arm are so obvious that the control arm is switched to the treatment. So I think it's a beautiful story. And basically, so the proteasomes, they can basically serve as targets for a therapeutic intervention in different diseases. And like I said, the bortezomib is used for the treatment of myeloma, and there are new proteasome inhibitors that are in clinical trials, and other proteasome inhibitors which are under investigation. And just for those of you, because you're living now in Israel, and maybe you learn a little bit about the healthcare system here, you know about the health basket.
Mhm.
You know about the health basket? Like there's a certain, if you have an HMO insurance, that there are certain drugs that you are entitled to get for not free, but for minimal amount of money. So the bortezomib for multiple myeloma or Velcade, it's in the health basket. And to get into the health basket, it's not a trivial matter. So I mean, it's a very robust drug and used in the clinic. And I wonder, some of you might want to exercise if you are going to the, how do you call it, the internal department, and they talk to you about the bortezomib. So, you know, find out that the doctors really know that it's against the proteasome, because sometimes when you keep on treating patients, sometimes you forget the molecular targets. So remember, we still have to deal with the ER-associated degradation. And what was the problems that we had with the ER-associated degradation? Remember that we have the abnormally folded proteins that we still need to dispose of, right? So if we need to have a degradation system in the ER, we need to worry about which problems, what needs to concern us if we want to dispose of proteins that are stuck in the ER.
Folding, how we get them out?
Well, okay, let's take it from 2 points of view. If we were to degrade them in the lumen of if these were luminal proteins and we were to degrade them in the lumen of the ER, let's say that there were proteases there, would that be a viable option? I mean, do you think that there's a problem here or not?
Selective.
Yeah.
If they're not selective, and remember what's happening in the ER, we're synthesizing, so we want to take very good care of all the products that we synthesize. We don't want to just degrade them, and we really need to segregate synthesis from degradation, right? We have to make sure that these processes are segregated. So we don't want to degrade so fast where everything is being synthesized. So the ER contains a quality control system like we discussed to prevent the immature or abnormal products from escaping. And the defective proteins or mutated proteins or proteins that are associated with certain disease, if there are mutations there, they will stay retained in the ER either until they're folded, like we were talking, for example, about the connexin cycle, or they are degraded, and this process of degradation is called the ER-associated protein degradation. ER-associated protein degradation. And what are the features of this ER-associated degradation? So it seems that it is associated, or it takes care, or it eventually degrades the substrates that are stuck in the ER. It's insensitive to lysosomal inhibitors, so it tells us that they're not degraded in the lysosome. And it also occurs in conditions where ER to Golgi transport is blocked. How do you think we can block ER to Golgi transport? Give me a general idea. What? Something that will inhibit the generation of COP2 vesicles. Right. So, so this degradation occurs somehow in context of the ER, but we still have to worry because we can't imagine that it's going to happen inside the ER. And again, the degradation should be extremely selective, and it has to be extremely segregated from synthesis. So how do we achieve that? So let's remember what we were talking about when we talked about protein transport into the ER. Remember, we had the transport, we had the translocon. Remember our translocon, where the protein was inserted and synthesis occurred in the ER, this translocon. Here we are, the translocon, the ribosome, and translocon and insertion of the protein. Now the beauty of the system, the unfolded proteins can also go out through the translocon. Tell me you're excited. No? Okay.
Well, what about proteins that haven't folded yet?
Okay, so there's going to be the system that will recognize that they still have a chance. Maybe they're in the Connexin system. Maybe they have additional chaperones. The question of when is something terminally misfolded is a big question, and I can tell you that a lot of labs are working on that question. How do you actually identify that it's terminally misfolded and you can't help it out? And in the break, what's your name?
Eddie?
Eddie came to ask me, how can we improve folding of a protein? And actually for different kind of folding diseases, such as, for example, CFTR, there are chemical chaperones that are under clinical trials that you can actually administer, and they will help the protein to fold better. So we can, in certain cases of disease, circumvent this. And the system that senses folding in the ER is more sensitive than the actual activity of an enzyme. That is, even if an enzyme is incompletely folded, it will get stuck in the ER. If we can help it fold a little bit and get out, it still may maintain certain activity. And then it will have residual activity, but it's better than none.
But like you said, if you help it fold a little bit, it's still misfolded. What stops it from being taken out by proteasomes rather than moving forward?
So the question is very valid and legit, and there is no one answer, and a lot of labs are working on it, and it's going to be different for every protein depending on what are the accessory molecules that are participating in its folding. And for certain proteins, you can improve that process by administering chaperones or by improving otherwise the folding. So going out of the ER can also occur through the translocon. Okay, so it's a kvish du-sitri, it's a two-way street, translocon, and also heavily investigated. Is this the only way out of the ER, or are there other designated streets that can take the proteins out of the ER? So the proteins are degraded. The proteins that are stuck in the ER are basically degraded in the cytosol, and people are asking the questions, and this is more mechanistically to what you asked. Is somebody from the cytosol pulling them, or is something from the ER pushing them? And what are the signals for this ER-associated degradation? And what are the molecular players? And what's the conformation of the channel? How can it play the two-sided roads versus one-sided road? And how does this occur? And this is under investigation. So now let's talk a little bit about diseases involving the ubiquitin proteasome system. So folding diseases, we talked about I gave you an example of immunofluorescence microscopy of the Perleus Meltzbacher associated protein. We're going to talk a little bit about the alpha-1 antitrypsin and about the CFTR, did you study about the alpha-1 antitrypsin?
Yeah.
Yes?
A little.
Okay, great, okay, good. So you'll see it now in a different context. CFTR is also, it's not Chinese, you heard about it before. We'll go back to that. And we'll also talk about mutations in the machinery. What happens when there's a mutation in an E3 ligase? And I'm going to give you two examples for that, which is Engelmann. Have you studied about Engelmann disease?
Yes.
Okay, great. And Parkinson. And did you know that one of the mutations in the Engelmann is an E3 ligase?
No.
So now you will know. Okay. So we're going to start off by the mutation in the substrates. And again, it's just examples because there will be more. And if you'll be, and when you'll be doctors, I'm sure that you may observe different other cases as well. So alpha-1 antitrypsin, this condition was described in 1963 following identification of blood samples that had a missing protein band in the alpha-1 region. And this takes me a little bit back to the concept of what I was discussing with you before. Remember, we were talking about glycosylation, the effect. We said that sometimes the analysis of blood samples can be very informative when we're talking about the systemic kind of disease. This was the case for the alpha-1 antitrypsin. Now, the finding was that several of the subjects had severe onset of emphysema, and it was found to be the biochemical studies showed that we're talking about the alpha-1 protein, which functions as an inhibitor of trypsin, and therefore it was called alpha-1 antitrypsin. At the same time, there were also animal experiments, and basically these kind of experiments and these observations were the basis of drug development for this particular condition. And when we're talking about the alpha-1 antitrypsin, so who makes the alpha-1 antitrypsin? The liver makes it. And what does it do, the alpha-1 antitrypsin?
Goes to the lungs.
It what?
It protects the lungs from.
How does it protect the lungs? What does it do? From the elastase, right? From the neutrophil elastase. So what's going to happen if we don't have the alpha-1 antitrypsin protein?
The elastase will be broken down.
The elastase won't be broken down, and then it will break down cells in the lung, right? Okay. So actually, the frequency of the alpha-1 antitrypsin is 1,000 to 5,000 individuals, and basically it will predispose to liver disease, and we'll see in a minute why, and also, like you said, to the emphysema. And when we're talking about the alpha-1 antitrypsin, so one of the mutations, or a series of mutations in alpha-1 antitrypsin, what is the outcome, the cellular outcome in the cells that are making the alpha-1 antitrypsin? Is it made, the alpha-1 antitrypsin, or it's not made at all? It's made. Where is it? Where do we find it in the body? Inside the liver. Where? Inside the cells. Where? In the ER. Why?
It's not conjugated.
Mutation which doesn't enable it to fold, okay? So we have, and that's one of the problems why we also have liver problems. Why do we have liver problems? Because the alpha-1 antitrypsin is clogged in the ER of the liver cells. So that's also going to affect the liver cells. Now this alpha-1 antitrypsin, while it's stuck in the ER of the liver cells, it will not be secreted, but it will be inside the cells, or it will be degraded by the proteasomal pathway, which we just discussed. So just anecdote, I don't know if it's true or not, but the internet said it, that Michael Jackson had alpha-1 antitrypsin for several years. Maybe yes, maybe not, I don't know. Anyway, so diagnosis. Diagnosis can also be a little bit complicated sometimes, because you're looking at situations where you can have chronic liver diseases, and it's going to be maybe in children as well as in the adults, and increase the risk for the emphysema in the adults. And because of this variability, basically this manifestation of disease can be found both in patients, both in adults as well as in pediatrics, and it can also cross or span a wide range of medical specialties. So sometimes one might think that we're talking about a very rare disease, whereas it might be more common than might be suspected. Okay, what kind of treatments might be available? So again, there's a substitution therapy, where one can get a substitution for the alpha-1 antitrypsin. And why is substitution treatment for alpha-1 antitrypsin so feasible, as opposed to, let's say, the CFTR, or what you asked me before? Yeah.
This is supposed to exit the cell to go somewhere else.
So
you can just give it to the cell.
So you can inject it, and it will find its way to the lung and protect what it needs to protect in the lung. You don't have to worry about it being inserted into the ER and then going to the membrane correctly, etc., etc. So you can actually give the drug. It's even easier than the Gaucher situation, because it doesn't need to be uptaken by the cells. It can just be in the plasma. Okay, so it's a beautiful example where the substitution therapy can actually work very nice. So eventually, of course, lung transplants, liver transplants, etc. And there's also studies on the idea of differentiating embryonic stem cells along a hepatic lineage, and then the liver cells can be directed to generate the alpha-1 antitrypsin properly. So this is just a list for you, of course not to remember, but to give you a feeling that there are different kinds of drugs that are being thought about, and several of them, as you can see, are also FDA approved, and several of them in clinical trials. All right, so now we're talking about mutations in the machinery. For example, the E3 ligase. So when we're talking about the Engelmann syndrome, it's a neurogenetic disorder, which I understand that you heard a little bit about, and it's characterized by the severe mental retardation, ataxia, seizures, EEG abnormalities, and bouts of inappropriate laughter. What happens with these Engelmann syndrome patients? They fail to inherit a normal active maternal copy of what? Of a ubiquitin protein ligase. Of a ubiquitin protein ligase, which is encoded by this gene. You don't have to remember the names, okay? So what's happening? We have the patients, they have 2 copies of the gene, but one copy is not wild type. One copy is mutated. Okay? So there's animal models in Drosophila as well as mouse models for Engelmann, and using all kinds of models like that. Okay, just for the incidence, it's 1 in 2,000, 1 in 20,000. I guess that you already know that if you've heard about this disease before. And I thought that an extremely interesting paper in that respect is a paper discussing the fact that since one allele is silenced, and if it's the normal allele that is silenced, then this particular paper talks about drugs that can basically activate the normal allele. Okay? So therefore, there will be basically a appropriate function of the normal allele as well. So just another example, and to tell you that the Engelmann syndrome, which we've already studied a little bit about, it's based on a mutation in the of a particular ubiquitin protein ligase. Parkinson's disease. There are forms of Parkinson's disease which are caused by a mutation in an E3 ligase, which is called Parkin, and those mutations are associated with autosomal recessive juvenile Parkinsonism. And what might be the substrates? And this again, it's a particular example, and please don't remember the name of the substrates. That's not the idea. But I do want to show you that this particular Parkin E3 ligase has more than one substrate. It has a series of substrates that it acts upon. And when this Parkin is mutated, it's associated with juvenile Parkinsonism. So how do you think that you can identify the protein consequences? What happens to the proteins in the cell when an E3 ligase isn't functioning properly?
Polymerization.
But E3 is not necessarily for ER proteins that are being degraded. E3, the question was a general one. How can you show me, or how can you design an experiment to figure out what happens when a particular E3 ligase isn't working? Sorry?
There's no specificity.
But what would you look for? Yeah?
You won't get ubiquitination?
On a certain set of substrates. So you can, for example, compare the degree or the pattern of ubiquitination between 2 cell types, one that is expressing the mutated E3 ligase and one that is expressing the normal E3 ligase, and you have means to label ubiquitin, and you're going to see what is the pattern of ubiquitination, and then you can get an idea of the cellular consequence, the cellular protein consequences of an unfunctional or of a mutated E3 ligase. Yes?
How would you determine if that ligase is ubiquitinating? Because I know some proteins are ubiquitinated by multiple ligases. So how would you know if it's from one or the other?
Well, if I'm suspicious of a certain E3 ligase that is not functioning, then I will set up an experimental system where I will isolate this particular E3 ligase. I will insert the defective E3 ligase into normal cells, and I will insert the normal E3 ligase into normal cells, and I will compare the pattern of ubiquitination. And I will see what is the pattern, and on the malfunctioning E3 ligase, I will see which proteins are affected. It won't necessarily tell me that these are the direct substrates, because it might be a secondary reaction or something like that. But the only difference will be plus the defective E3 ligase minus the defective ligase, okay? And again, when we do experiments, we're only allowed to have one variable, right? So the one variable is the E3 ligase. Mutated, unmutated, what's the difference? How can you identify binding partners, so ubiquitin protein ligase? How can you identify substrates, for example? You can invent, you don't need to actually go to the lab to do it now. So let's say you had a ubiquitin protein ligase and you had some sort of cell extracts, right? You can look for binding partners in the cell extract and identify which protein bind to that. And we discussed basically the identification of ubiquitinated substrates when we talked about the defective E3 ligase. And there are all kinds of biological processes that are affected by ubiquitination. For example, the skeletal muscle wasting when we fast or when we starve or when we're denervated, or even for, you know, when we have a cast on our hands or our legs, when the cast is taken off, often the leg is thinner, right? No?
Yeah.
So often that has to do with ubiquitination processes. Okay, so again, the specificity is achieved by the ubiquitin ligases, and basically they're going to be able to recognize different primary and or secondary post-translational motifs in the substrates, and a wide array of modifying enzymes such as kinases and chaperones, which modify the substrates and will render them susceptible to ubiquitination and eventually proteasomal degradation. Now there's additional modes of regulation, for example, cellular localization or the nature of the ubiquitin chain. Ubiquitin can be added to a protein on different sites, and also the stability of the ubiquitin chain itself. So let's say a polyubiquitinated chain is added. So we have ubiquitinating enzymes. What are we also going to have? Which kind of enzymes? Deubiquitinating enzymes. So another mode of complexity. Ubiquitin is not only added, it can also be removed.
What's a situation where you would ubiquitinate something and then decide, no, I don't want to destroy it after all?
It's not a matter of destroying. Sometimes you ubiquitinate for signals, which will not necessarily mean that it's immediately degraded, and you might want to do an initial step of ubiquitination, because if you have more than one E3 ligase, you can think about a complex situation where addition of the ubiquitin chains is added by one E3 ligase, and the second ubiquitin is added by another E3 ligase, and so on and so forth. Complexity is almost infinity, as long as you remember the basic rules: E1, E2, E3, substrate, and signals that are going to govern it. So basically also ubiquitin tagging can function other than in degradation, and you may hear a little bit about that in the lecture on endocytosis. Sometimes you get monoubiquitination, and that is going to be associated with targeting signals. And sometimes if you have a region which could be ubiquitinated, sometimes you can get monoubiquitination on more than one site. Now, ubiquitin is basically added to lysine residues. Ubiquitin is added to lysine residues, classically added to lysine residues, but it has been shown that ubiquitin can also be linked at certain cases, in certain cases, to the N-terminus of the target protein. But again, classically, it's targeted to lysine residues in the proteins. And how do you think that polyubiquitin chains are formed? How does ubiquitin bind to ubiquitin? I gave you the answer in the previous sentence. How is it? Through lysines, okay? So lysines of the ubiquitin are going to be the sites that are going to be ubiquitinated, or the binding site of the following ubiquitins.
Is it a cooperative binding?
What do you mean by cooperative?
Like, does the binding of one cause more binding?
It's a matter of signals, and it depends which E3 ligase and which substrates, and everything depends, and depending on the signal, etc. Okay, so again, another mode of regulation, because ubiquitin itself has more than one lysine, depending on which is the lysine in the ubiquitin that is forming the chain, that can also add degree of complexity. So we have ubiquitination. We also have deubiquitination, which can regulate the length of the ubiquitin chain. And we talked about the fact that ubiquitin can be added to different linkages. There are also ubiquitin-like proteins. We're not getting into this, but that is the case. Now, if I asked you, how do you determine the mode of degradation of a certain protein? So you will tell me, rightfully so, that we can use specific inhibitors, right? How can we inhibit lysosomal degradation? What would be the principle of inhibiting lysosomal degradation? How would we inhibit lysosomal degradation, assuming that the substrates arrive in the lysosome? How do we raise the pH? Raise the pH will inhibit lysosomal degradation. We have specific inhibitors for the protosome, and again, those are used in the clinic, and I showed you those. Now we can, if we think that degradation occurs through a certain protease, we can also mutate that particular protease and see if that affects the degradation of a specific protein. Yes?
Could you also, instead of just raising the pH, could you also stop the lysosomal hydrolase from even getting to the lysosome in the first place?
What, to inhibit the mannose-6-phosphate addition?
Or somewhere else in the pathway, just to stop the enzyme from even getting to the lysosome so it wouldn't be able to function.
But the lysosome has so many enzymes there that it would be hard, I think, to target one. And basically, this is more like for experimental strategies. How can you prove that a certain protein is degraded in the lysosome? And the classical measure for that is raising the pH of the lysosome. What is a dominant negative ubiquitin molecule?
And all of them are blocked and then the active one.
What?
All the
Mutation which changes the ubiquitin, so it can't be.
So it can't be what? It can't be tagged. Okay. What kind of mutation do you think would be a dominant negative ubiquitin? What won't it have?
Lysine.
Won't have a lysine because then it won't be able to generate the ubiquitin chain.
So you'll get monoubiquitination then? Because
You might get monoubiquitination,
okay.
but not necessarily serve for degradation. So there are also model systems like cell lines, which is defective in E1 enzyme. Can this be a permanent defect in the E1? What will happen if it will be a permanent defect?
Not viable.
Not viable. So what kind of defect it has to be? Some sort of inducible. Take temperature sensitive. And we have the yeast where we studied that they're easy to manipulate genetically. And we also talked about the in vitro model systems for which the Nobel Prize was granted for a discovery of the ubiquitin system. And with that we complete, almost on time, yeah? It's okay? And we meet on Sunday for cell cycle. Sunday, yes?
Yes.
What?
Yeah, Shibata.
Yeah, I kept hitting it and having to move it back into place. Sorry.
What's that?
I kept hitting the cord and having to move it back into place. So,